From the bioinformatician point of view, we like to be valued, but we keep working hard to be costless. So we try to program everything just to lower the cost more and more. So what I will try to do today is to kind of bring you an overview of the simplified tools that from the bioinformatics we have programmed to simplify all the data analysis for NGS resistance testing. Okay, so for an outline of this presentation, it will be divided in two parts. First is how is bioinformatics used nowadays for NGS drug resistance testing, how an ideal tool for data analysis should look like for the clinical application. And the second part will be basically an overview, a very oversight view of these 5 tools that are available today, and also talk a bit about the different efforts that are being done in standardizing all the analysis and the outputs in order to be able to evaluate these outputs among the different tools. Okay, so first of all, we like data. And these machines, as Martin showed just a bit ago, generate lots of data. So I try to put the pictures in kind of a relative scale on their size. It's relative, and it's not metric. But anyway, you have the high-seq, the MI-seq, the mini-seq, the nova-seq, the high-seq, the next-seq, you name it. But they all generate lots of data. This is the PGM from IonTorrent. This is the Proton from IonTorrent. And there you have the nanopore machine. And there you have the even smaller sequencer. There are some prototypes around already that fits into a cell phone. And you can both sequence and analyze from the cell phone. Okay, and also you have the Sentosa platform, which is most possibly a good choice for the clinical lab, but it's not in the scale in this one because I would need another screen. But anyway, all these machines generate lots of data. I mean, with Sanger, you know, you're used to couple of 3 sequences that you basically edit with BioEdit with any of these programs. With NGS, we always generate from 50,000 to 100,000 to 200,000, depending on the design that you have. So that's as Martin explained before, some bioinformatics is needed, and you need to when basically thinking about adopting NGS technologies, you also need to have some option on bioinformatics nowadays when incorporating these technologies. So why do we need that? Because we need to ensure that big data is of good quality, generates good quality reports, generates good quality information. We need to ensure that the data analysis is adequate to the data from different perspectives, not only for the quality, but also on the computational scaling that analysis needs. And also try to just, since we are starting with this NGS data analysis for not starting now, but some years ago, relatively short time, try to ensure that the results of this analysis are kept in a structured data way for exploitation in the future. Okay, so this is just 2 questions. We were circulating a poll on how different research labs were using NGS for genotyping HIV in Europe. This is I'm not sure whether this is really representative of the options of the reality, but it already gives a kind of a picture of how NGS is used, and mostly all reporting centers reported that they were used in a high-seq, MI-seq, you name it, seq machine, and that lots of these centers were also using some in-house protocols to prepare the samples and the DNA libraries. Okay, and again more than half of the labs were using an in-house bioinformatic pipeline, which is very common these days to use those kind of pipelines, because these research labs have the option to have a bioinformatician or bioinformatics support nearby, and they can use it to analyze that data. Okay, there were some that were already using genome-to-phenome, PASEC. We will talk about these tools later. But anyhow, what are the challenges for broadly used software or pipeline for this data analysis. Obviously, you need to make sure that your tool, from the computational perspective, can scale up. You have enough resources to analyze 1 sample every week, 10 samples every week, or 1,000 samples every week. You shouldn't be short of computational resources. That's one of the issues. Also, as a developer of one of these pipelines, you face lots of experimental designs with different features and characteristics that maybe can hamper the analysis if you tune your analysis too much to one experimental design. It may not be fit for other. So you also need to keep all the information on how the analysis has proceeded, where the errors were, if there were any, how the results were at every step of the analysis, and keep track of that and ensure that this analysis is reproducible. You also have to deal with the different chemistries that we have just seen. Sequencing chemistries, they all generate different error profiles. They have their particularities, and they affect how the analysis results can be produced. So also since there are so many different pipelines, so many different in-house pipelines, all the analyses are quite heterogeneous. So the strategies are heterogeneous, and the results usually are, you can compare them, but the comparison needs to take into account the differences also in the way that these analyses are running. And also the format and the interoperability of this data is an important issue when you want to compare these methods or to evaluate the accuracy of these methods. And last but not least, because this is a very important issue, is how I'm not talking about this, but I'm aware that this is an important issue. It's the data ownership and all the regulatory aspects of the data, because we are generating tons of data, and this data may be useful in the future for many epidemiological studies. But if you plan to open a pipeline in the public, you need to make sure that these regulatory aspects are taken into account. Okay, that includes for how long will you store the data, because you need to store it at some point, and who will be able to access that data. The most conservative thing to do is only the user can access the data. That's what we do. But maybe it's very good to think in the future some kind of policy to open the data, at least partially, to some consortium or some other program. So these are difficulties, complications when you need to think about when designing or programming a pipeline. But it has a lot of benefits as well. This is a diagram showing the kind of an ideal image of how NGS and bioinformatics could play a role in the future of drug resistance testing. Nowadays, that probably more and more testing will be needed here in Europe, but also in low and medium income countries, where the doctor or the clinician will send the sample to a central lab. The sample will be prepared with these protocols, either automatically in high multiplexed library preparations or smaller scale labs. And this data will at some point be uploaded to the computational cloud, where the bioinformatic tools will be waiting for it. And this bioinformatics will be simple to use, have a lot of features that will make it useful and adequate for this analysis. It will be usable only with a click, secure, highly scalable, with quality controls embedded, and all the resistance interpretation integrated within the tool. From this bioinformatic tool, the output will come out and will follow two separate paths. One will produce an individual drug resistance report for the clinician that will go back to the clinician and will enable some trigger in the clinical practice. And the other path will basically store the data, the authorized data, into a structured database where it will be useful for further real-time monitoring of the quality of the lab, of the emergence of any resistance or the file, or using this data to construct phylogenies, for instance, and will be able to inform authorities or officers on the program. So the benefits are very potentially high. So that we need to aim high, and then we will go down. But for that, we need that the bioinformatics tools, that piece in there accomplishes several features. The tool must not need bioinformatics support on the side, because usually there is none. And it may be a handicap for a clinical center or for a diagnostic lab to the need of hiring someone who knows about bioinformatics. It's maybe a problem, and it's a cost for sure. Usually these sites don't have computational support available, or it's very, very scarce. It must be usable remotely in a friendly manner. So as a bioinformatician, I know that most users don't like black screens or command lines or things like that. So you need to design a friendly web interface, because this is part of the way that you can reach the final user. You need to provide this robust analysis and easy to interpret results from the clinical side using well-established interpretation rules like Stanford, for instance. You need to incorporate the quality standards within and in a seamless manner. You need to avoid, because we are talking about large data volumes, and network may be not accessible. High throughput network may not be accessible in all the sites, so that's an issue. So we need to avoid unnecessary transfer of large data volumes and provide that information for download that it's useful and it's essential, like a PDF file, which contains the information. As I said, the computational infrastructure should be avoidable, an avoidable issue. And as I said before, it should be the bioinformatic ideal tool should respond to in a highly scalable manner to a virtually unlimited number of samples. And as I was saying before, we should translate into no cost or a minimal cost into the diagnostics lab once the data is available, especially in low and medium income countries where they may need to do large number of resistance testings. We should not be adding more cost to that testing. So this is a list of softwares that are available for NGS data analysis for HIV specifically. Some of them are commercial. The part on the bottom these are commercial tools. We have there the SmartGene, DeepCheck, and also the Vela Sentosa, which is the full pack, not only bioinformatics. And on the top, we have all these tools that have been available, some of them for a while. And either they need some or a lot of bioinformatics. And the three in the middle are those that they have a web interface, and then they can work essentially from the very beginning, the very raw files that come out from the sequencer. So they can basically, you just drag and drop the files from one place to another and click a button, and all the data processing comes through. Okay, so this is what we want to avoid, particularly someone sitting there as well. Okay, it's too many screens, it's a lot of space in the room, and you cannot see the sun. And also, this is a Linux station, so it's scary. So that is what we want to avoid. These three pipelines, as I said, need no processing of the data outside of the tool at all. And the good news here is that they agree on the results, mostly a bit less when they go down to the frequency of the mutations that they find. Okay, and now we go how do these pipelines analyze the data? First of all, we need to introduce some common concepts. I'm not sure where you are familiar with them. So all the sequencers that we have seen produce FASTQ files. This is the same as a FASTA file with quality values. So you have the ID of each of the sequences, the sequence of the read, and at the bottom of these 4 lines you have the quality encoded in some scale. Every letter there, you have the E, F, C, all these letters are the quality values of every one of the nucleobases. So the quality values is the number, which basically is a translation of the probability of having an erroneous call on that particular base. It goes from 1 to 40. The higher the value, the higher the quality. So the less the probability that you are making a mistake calling that base. Also, you have the pair-dense single-end designs on the experimental side that are affecting. And usually you use these quality values to filter all the bad reads out, or keep the good reads, again, good reads, in. So to improve the quality. Okay, so if your reads are in between the 30 and the 40, you are quite okay. And you have some chance that some of your base calls is wrong, but it's considered to be acceptable. Okay. Other than that, you have your sequence alignment, or usually in the SAM, BAM file formats, which is basically containing all the information on where each of those reads is aligning respect to a reference, which is called usually HXV2 or consensus B or whatever. And the depth of coverage, this is also a common concept that we use quite a lot. Means how many times a single nucleobase is read. So, for instance, if you have a 10,000 depth of coverage, you are reading every one base at 10,000 times. Okay. So this is the typical. Maybe there are some variations on that, but this is the typical processing of a raw file from NGS machine. You first do the quality filter, control. Once you have the good data, you align it against a reference. Conceptually, it's the same that you did with the Sanger sequencing, but with much more sequences and much more demanding. You can optionally take this alignment, which is produced in this step for further quality control. So you discard all the sequences that have been aligned in a non-reliable way, or do some other additional step like ApoBack filtering. And then you can use this alignment either for calling a consensus, which would be Sanger-like sequence, or do the variant calling and obtain either an amino acid frequency table or a codon frequency table, where every position you have the frequency of every codon that has been read at that position. And with that, you can go you can use that information already to do some interpretation on your preferred algorithm, with your preferred algorithm. And when you have this data, you can store it in a structured database, or you can simply throw it away if you want. But it's good if you can store it for further use, or for some time for the user to have it available. And after that, you can use that information. That's the ideal thing that we could do with this information, is to implement some nice real-time algorithms in these databases to study the clustering of new sequences that are coming in with some of those that are stored in the database. So the quality control is basically throwing out all those sequences that are bad or too bad, with a threshold that you can define. And the contamination control is another issue that may be affecting your data. There are two types, which is external. One is external contamination. Mostly, in my experience, it's human DNA contaminating the samples. You can have 400,000 sequences and 390,000 are human DNA sequences. Then you have a problem, because both you are throwing your money and you cannot really use your data for resistance testing. So it's not that you will be cheated by your software, because the software will say, I have no HIV sequences, I can do anything. But you will need that information to optimize your protocol. And the other is the cross-contamination, when during the library preparation or the sample prep or even the sequencing, because of the barcoding, you may be confounding one sequence from one sample to another. This is a plot that we generated with an independent software showing, I don't know if you can see it clearly, but all these colored lines are transfer of sequences that are assigned to one sample, but if you process them and try to assign them using similarity, they go to another sample. So you need, this can create artifacts and miscalls in the resistance, particularly if you go down with your thresholds. And it's better if you can remove this cross contamination. Okay. So sorry. Now as a second part, I'm going to go to these 5 different tools. I think that since there was not a chance to play with them here, there was not the time either. So it's better if you play with them. I strongly recommend that you just register to these tools, every one of those. I have fun with those, but it's a way of learning how they are very simple to use. They have free examples, and you can try it. So for starters, we have the Gino to Fino, which is developed at the Max Planck Institute by Matthias Dering. There is a poster here explaining number 25, I think. And so this tool is free to use. You can just upload your data, and it provides a report. I think we have seen some presentation as well with examples. And it has been, the paper associated to it has been recently published in the Nucleic Acid Research. And it basically provides some resistance interpretation at different levels of detection for each of the available drugs. Okay. This tool creates this report. You can download the reports or in PDF formats, which is very handy if you want to send the results or print them. It detects ApoVec mutations. I think I don't know who mentioned. I think it was Alex. You mentioned that the ApoVec mutations are detected using the approach by Bob Schaffer using codon signatures for ApoVec activity. This tool needs a codon table as an input or an amino acid table as an input, so some pre-processing of your sequencing data is needed. And you can also define, it uses Gino to Fino resistance algorithm for the interpretation. It's maybe not so common because other tools tend to use Stanford. And it doesn't need any registration. And the good thing is that it also accepts HCV data. A second tool is, I don't know how to pronounce that, Hyrax X-type, this is developed in South Africa from Dr. Simon Travers. And it's on all the development that his group has been doing on software and NGS data analysis. They have produced this kind of a spin-off. This is a commercial tool, although they provide some free testing for some samples. This is very easy to use. You register, you upload your data. Either you can upload one sample or many samples at a time. It's very fast. You can define your cutoffs. Just press a click, and you obtain your results, number of reads that are passing the filters, number of reads that have been discarded because of low quality, if they are paired, if they are forward or reverse. You have the coverage plots. We have seen some for all the samples, the median, the mean coverage in your set of samples as well for quick tour of your data quality. This is very, very handy as well. And then you have all, for every of the samples, you have your output, your result on a defined threshold. In this case, it's a 15%. You have the interpretation of resistance for every of these drugs that you can also export as a PDF format and send it or whatever. So this is cloud-based. It's commercial, as I said. It has an embedded all-in-one quality control. It does the sequence alignment, the variant calling. All the steps that we have seen are done by this tool, and it uses Stanford HIVDB for interpretation. It's highly multiplexed, so you can upload the data for one sample or thousands. Well, thousands I have not tested, but for 20 you can. And can also accept data from Ion Torrent 454, if you have any, or Illumina. And a good, very technical feature of this software, I think, is that it's using an aligner that is codon aware, that has been developed in that group. And that means that the insertions and deletions will be better treated with a codon-aware aligner. And you can try it at this URL here. The third tool, I think Richard is somewhere there. So this is developed at the British Columbia Center for Excellence in HIV. It's embedded. I don't know if you are aware of Illumina Base Space. Illumina Base Space is a web platform that's provided by Illumina if you buy one of their sequences or if you just register. And basically, they have hundreds of different web applications that can analyze your data, either for drug resistance testing, either for exomes or cancer or whatever, you name it, or just building bacterial genomes. And the good thing is that this tool is embedded in the good thing that the fact that this tool is embedded in base space, that you don't need to transfer data. That the data is in base space, so it's basically internally transferred, but you don't need to download it or upload it. And you can use the raw data as input. Also, a good technical feature is that if you provide your control reads, which are not HIV, you can assess for every of your runs an error model which is used to correct erroneous variant calls and improve the quality of your data. You can download all your intermediate files, but you need to register to base space. It's supposed to be free to use. Yesterday I logged in. It's 3 iCredits, which is more or less $3 for sample. But I know that it's going to be free because it's the main purpose. But I think there is some Illumina interest there, so I'm not really sure. And also you can also download the reports and print them as a PDF, and it's also one of the features that is common in these cases. There we have the Hydra Web as well. This is developed by Eric Enns at the Public Health Agency in Canada. You can also register. It has upload many samples at a time. You have lots of parameters to tune your analysis with very reasonable defaults if you are not interested in doing that. But these are controllable, and that's a good thing. You also can see the coverage, for instance, for many samples at the same time, and see how the general quality of your run has gone and how it looks like, and discard any sample if you like. Sorry. And you can also download the intermediate files, see how the qualities have gone, how many reads have been discarded, or if there are enough reads, and play with the threshold that you want to use to interpret resistance. But the resistance is provided as a table saying if the resistance is if the mutation is resistant or not, but not to which drug, only if it's an RTI. So the most common, I guess that everybody knows to which drug they are resistant, but the others maybe not. So in this case, there is no PDF report that, as far as I have seen, and you can also play with this tool at this URL. Okay. And finally, the last tool that I'm going to talk about is PASEQ, which is the polymorphism analysis by sequencing. It's also available at this URL there. You can register and log in. It has also a very intuitive user interface where you basically drag and drop or upload your files, either through your computer or with a Dropbox link or you name it. And once you have uploaded data, you basically press a button and you start the analysis. When it's finished, you have all the samples there, basically resuming all the resistance that have been found in these samples, and also the subtype. And you can get into every one of these analyses and have more detailed information on the processing, all the logs on the processing, all the quality filters that have been applied and how they have improved the data, all the proportion of contaminating reads, if any, in that sample, the proportion of low quality reads that have been discarded, and the remaining reads that have been analyzed, and the coverage shown for every one of the proteins in the HIV genome that you have sequenced. Aside from that, obviously, you need a report for every one of the drugs. You can play also with the threshold there. at 5%, at what percent, and you see the changes in drug resistance of the virus in real time. You can also plot in similar way the geno to pheno. All the resistance information is plotted into a backbone, the WHO backbones, so you can easily spot which of the recommended drugs are have some resistance in the virus or not. And you can also export all this information into a PDF file that can be easily printed. Okay, so specific features of this software is it also detects ApoB mutations, but it doesn't use the approach by Schaffer, but it uses the hypermute algorithm to detect ApoB edition. You can also download all the intermediate files. It's free to use. It's also cloud-based, and it's highly multiplexed as well. And it also has some sharing options to basically open your results to particular other users that you want to share your results with. And finally, just a slide on the efforts that are being done on the standardizing the analysis and standardizing the outputs and the formats. The first one is from the Winnipeg Consensus, which is led by He Zhaoji in the Public Health Agency in Canada, in which we participate as well. It was a symposium that defined some recommendations, some suggestions on how the future pipelines for analysis should take into account for how the analysis should proceed in those pipelines. So this is submitted now. It will be published sooner or later, I guess. Sorry, this is too close, but also from that symposium and that consensus, it was suggested to come up with a variant data format to be able to exchange all this information among different pipelines. And it was based in the HMCF format by Eric Enns, also in the Public Health Agency in Canada. It's applicable to HIV, so it's being defined now. It's applicable to HIV, but also all other genomic populations, in particular viral populations. And the idea of this format is to basically overcome all the limitations of collapsing all the NGS information into a single consensus data sequence, because you lose resolution and you lose information. And also there is in development this dry panel from BQA. This I think it's 12 samples, I think, distributed among different centers. They have been sequenced independently, and all the analyses have been, and all the files are being analyzed by different pipelines, and then all these results will be compared in order to reach a consensus and to see how the different pipelines and different labs perform in extracting the mutation information and the resistance information. And I think that's all from my side. Just to thank you all for just hearing me. And also the group and the microbiome, and Toti, who is the web programmer in Lluita, who is helping a lot in all the design and all the programming of the web. Just thank you.